हेलो हेलो मंजू। हेलो विनय।
Yes, sir. Online Zoom. It's YouTube as well as we have students in the other room. Okay. Yeah. It's very. Yeah. Good morning, everyone. I take the privilege and pleasure of, on behalf of entire Indian Institute of Horticultural Research, we have the privilege of hosting Professor Indranil Dasgupta, who is currently INSA senior scientist in University of Delhi South Campus. He is based in the Department of Plant Molecular Biology. You may all know that Department of Plant Molecular Biology was the hub of genome sequencing several years ago. That's where the rice genome sequencing started in collaboration with IRI and other partners at the international level. So before I start introducing our invited speaker, I request our respected director to yeah, welcome, Professor Dasgupta. Sir, we have a small moment. I also take this opportunity to welcome our director. Respected Professor Indranil Dasgupta, invited speaker of today, and respected director, sir, and all my colleagues. I would like to briefly introduce the speaker today. Professor Dasgupta did his undergraduate studies from Presidency College in Kolkata. It's a very famous college. Then he did his masters and doctoral studies from Jawaharlal Nehru University, that is JNU, in the biotechnology department under supervision of Professor H.K. Das. In the early generation, that was the lab which had the first genetic engineering experiments initiated in the country. That's where the thing started. And after his PhD, then he moved to John Innes Center in UK, which is based in a city called as Norwich, which is one of the top plant science research institute in the world. So he worked with a scientist, famous virologist known as Roger Hull for almost 6 years, to start with as a postdoctoral fellow and then as a research scientist. And after returning from John Innes Center to India, he joined Tata Energy Research Institute, that's TERI, along with Professor Deepak Paintal, Professor Anil Grover, and several stalwarts of biotechnology. So they were there as a group, and eventually all of them moved to Delhi University South Campus in different capacities as professor and associate professor. So since then, Professor Dasgupta has been working in Delhi University in the Department of Plant Molecular Biology. He has guided several students. He had several national level and international level projects, and I was lucky to be his student. And I worked in a project that was funded by European Union, and I had the opportunity to avail DART scholarship and visit Stuttgart University as well as John Innes Center during that period. So we had a lot of freedom. There was that was the beauty of the lab. We had a lot of freedom to work on our ideas and come up with good publications. So then he has guided many students, and I don't want to tell much. He is recognized. He has been vice president of Indian Virological Society for a couple of years, and later, after while being professor of in Delhi University, he also served as dean of South Campus for a brief period. And then he was having J.C. Bose Fellowship, one of the prestigious fellowship during fag end of his tenure in South Campus. And now he is INSA senior scientist. He is superannuated from his regular services, but however, he is continuing pursuing his academic interests, especially in the field of virology as INSA senior scientist. This is also a very prestigious position. And he is a recognized fellow of all three different academies like Indian Science Academy and National Academy of Sciences, Allahabad. So he's very well recognized, and currently he is visiting different institutes in Bangalore. He also has a meeting in Indian Institute of Science, hosted by Indian Academy of Sciences. So we had the I came to know that he is going to be here, and I thought we should make use of this opportunity to invite him to know more about. He has done a lot of work on viral promoters and has several patents, including an US patent and Eurasian patent. So he will explain in detail about plant viral promoters and any other important areas of plant virology. Sir, with your permission, you want to say, yeah.
Yeah, so thank you, Professor Indranil Dasgupta ji. It is really a good opportunity for all of us because to all of the head of the divisions, my scientists, on behalf of all of them, I again welcome you, particularly for this institute. As I mentioned, this institute, we are working mainly on horticulture crops, particularly the fruits, vegetable, flowers, and medicinal crops are there. We have a different experience in the plant system as well, in the viral other things in plant pathology and virology. So I think it is a very you have a lot of experience. I know that when I was in IRA, I came to know from the Dr. Anupam Verma. So you are working in the way of in cucurbits virus. 28 virus in one crop. It is there in case of cucurbits. When I work in cucumber and bitter gourd. So that sort of things we have. So we have very good interactions with the all virologists there, including, as you mentioned, Dr. Mandal and other people, those are working there. So I'll not take so much of time. As you mentioned, it is a wide experience he's having in the transgenic virus, then viral resistance, and also many of this work he has done, particularly the viral promoters is concerned. I think it will be very much of enlightened to all of us, definitely, sir. And we hope that all the students also they joined in other also here in another one seminar hall, because this is a small one, and some people also joined in the online. So again, welcome all of you, particularly listen and have some doubts or queries, please ask, because he's almost a teacher. So the teacher wants that it should be an interactive meeting. So once he is giving this lecture, I think we can also learn many things from him. Also, we have what the experience, what he has to share us, and we can also have some interaction with you. Thank you, sir, for being here today. Thank you.
So I can use this.
Yeah. This one also.
Okay. Good morning, respected Dr. Behra, Director IIHR, Dr. Patil, and all my other colleagues and friends. So, yeah, it's a great pleasure visiting this institute once again. I've come here 2-3 times earlier when Dr. Krishna Reddy was here in plant pathology, and I'm very impressed by this institute's work and a lot of potential it has created. So today I will be speaking mainly on how we have worked on the promoters of some viruses, which we tried to use it for expressing interesting proteins and other molecules. And then I will try to speak something on how we have used plant viruses as vectors for expression and gene silencing. And finally, how we can use plant viruses or plant virus derived vectors for gene editing, which is something which is coming up now. Okay, so that is the topic. Plant viral promoters as tools for controllable gene expression. I will not talk about resistance aspect at all, because I'm just focusing on this using the plant viral promoters. So can we have the next slide? Or I can use this? Okay. Oh, okay. Okay. No. This one. Yeah. Yeah. So these are some of the points which I will try to cover. Indian scenario for plant viruses. Again, this is a very general topic, and I will be focusing on promoters of 3 or 4 viruses. One virus infecting rice. I know rice is not a mandate crop here, but I think one can extend some of the information which we get. And viruses affecting cassava and okra. Cassava project we had for several years. Dr. Patil was also in our group at those days. And okra project, we had a very small project on okra funded by BIRAC. BIRAC is a agency under DBT. And then I will talk a little bit about gene silencing vectors for rice or cereals and virus-based gene silencing in horticultural crops. I don't have too much of information, but I'm sure with all the experts here, it will be useful. And also, finally, virus-based gene editing, which is, I think, very emerging and very useful biotechnological tool. Why viruses? I don't think I have to spend too much time on this. It causes variety of diseases in tropical countries such as in India. Losses of fiber and food production is very significant. Most importantly, which is more relevant to this talk, is that viruses lend themselves easier than other pathogens. When you compare viruses with fungi or bacteria, viruses are much more easier to work on as from the biotechnological perspective. And most importantly, viruses could be used as starting points to develop gene expression, gene silencing, and gene editing systems for plants, which is emerging these days, which is the knowledge is emerging these days. This slide shows a few of the important viral diseases of India. Again, it is not an exhaustive information, but we have viruses of legumes, mung bean, etc. Okra, in fact, there is a lot of work which was done by Dr. Krishna Reddy and Venkat Ravanappa in this institute on okra viruses. Cotton viruses, cassava viruses, these are important things. And tomato leaf curl, of course, is also of interest to this institute. And pigeon pea sterility mosaic, Dr. Patil has done a lot of work. Groundnut bud necrosis, papaya ring spot. So these are some of the important viruses affecting crops in India. And we can have a lot of, there's a huge amount of information already available and generated by Indian scientists on these viruses, which can be used. This is a very old slide, and this was published in 2003, I think, on the effects of yield losses. Now, yield loss, calculating yield loss is not easy because under field conditions, you always have multiple infections. But you see some of the yield losses are very significant, especially for pigeon pea or tomato, and even for rice, the virus on which we were working. It says 10% loss, but if you look at the importance of rice, it becomes very important. And these are different types of viruses belonging to different viral groups. Okay, and this slide, I don't know whether you can read all of them, but this slide I prepared recently. Viruses being studied in India. Again, there is no real database on who is studying what virus in India. But I relied on these Virocon meetings. I have taken this information from Virocon 2023-2024. So Virocon is a virus conference organized by the Indian Virological Society every year. And I've taken this information from the abstract submitted for poster and oral presentation. So what we see is that there are about 15 viral families which are being worked on in India. And interestingly, there are about 32 viral families known, plant viral families known. So what is interesting is in India itself, we are working on more than half of the families of plant viruses known all across the world. And there are about 68 viruses included in this. So what you see is that mostly there is a large number of Gemini viruses were being worked on in India, and there are about 24 Gemini viruses. And then there are potiviruses and other colimoviruses, etc., etc. So what I'm trying to say is that all this information and the expertise and the material, cloned RNA, DNA, genetics, and vectors, and isolates, they are all present within the country. And what is important is maybe agencies such as ICAR or DBT or ANRF, they should sit together and try to utilize this resource. So that is my humble request for planners and all that. Okay, so next slide. Again, these are sort of classroom type of slide. This is the life cycle of a DNA virus. So DNA virus, when we are talking of, we are mainly talking of Gemini viruses and Badna viruses or Tongro viruses. So they usually, Gemini viruses are transmitted by whiteflies, and they get into the DNA, the nucleus of the plant cell, and they undergo replication and transcription. What I'm trying to say here is that circle in the red, viral transcription machinery can be exploited to drive proteins of interest. So we can use the transcription machinery and the transcription signals to drive proteins of interest, and maybe sometimes even RNA or DNA of interest. No, I can. It's okay. So finally, what I want to say on the right, to the right hand side, is the Badna viruses and Tongro viruses and their close relatives, colimoviruses, contain double-stranded DNA and have been used as sources for promoters from the dawn of plant biotechnology. What we know is transgenic plants started to be made sometime in the late 1980s. And from there, from day one, plant viruses from colimoviruses, CMV35 as promoter, has been used. So plant viruses have played a very important role in plant biotechnology, there's no doubt. The next slide is the RNA viruses. Most of the plant viruses are RNA. And there's no clear reason why. But plant RNA viruses, they don't enter the nucleus, or they don't directly enter the nucleus, and they undergo translation, and depending upon whether they are positive sense or negative sense, there are some differences. This one, what I'm showing is a positive sense RNA virus. Most of the plant viruses have this strategy. So there is active translation and transcription. Transcription in the sense there is a replicase, viral replicase, which multiplies the viral RNA. And there are quite often subgenomic RNAs, etc., which are also shown there. So again, these viral replication or transcription machinery can be exploited to drive proteins and RNA of interest. And this will become this will become relevant when we discuss viral induced genome editing or gene editing, where these have been used very extensively. And also because they are not entering the nucleus, and they are probably not transmitted to the next progeny, they are considered to be safe, because otherwise they may sometimes integrate into the DNA. So, but there are some issues which we will discuss later. Okay, so the next slide, yeah, I will just talk about these four viruses. Although we have worked on more viruses, but for today's talk, we'll just discuss these. So one is rice tungro bacilliform virus, which we have worked, we started working almost 25 years back. It is a family Colimoviridae, and okra ination leaf curl virus, Sri Lankan Kassav mosaic virus, tomato leaf curl New Delhi virus, they all belong to Gemini viridae, single-stranded DNA. Tomato leaf curl New Delhi virus, we have not done anything significant, but we have used it as part of our study. And we have used the promoter elements of the above four viruses. They have been also, some of them have also been used for gene silencing work. So first, then next few slides are the rice tungro bacilliform virus. It causes a disease quite extensively in our country as well as neighboring countries of Southeast Asia. So yellowing of rice and all. I mean, fortunately for us, this virus has not appeared in our country for in the last 10-15 years. So it is, it is sort of, it is maybe lying dormant. We don't know, but there was one publication in 2022 from Bangladesh, and that area, Bangladesh, eastern Bangladesh, Kumilla district, that is absolutely adjacent to Tripura. So because these are spread by leafhoppers, so they can always come back. Anyway, so this is a DNA virus belonging to family Colimoviridae. So in the next slide, I'm just showing the I'm just showing that these viruses, RTBV, is found in two groups. One is found in India, our country, and in Sri Lanka and Bangladesh. This is called the South Asian group, and Southeast Asian group via RTBVs are found in Philippines, Malaysia, and Indonesia, etc. And that table on the right shows the nucleotide similarities and the amino acid similarities between the two groups. Both of them are rice tungro bacilliform virus, but there is a significant difference between them. And we started this work in early 2000, and the idea was to study the Indian isolates of rice tungro bacilliform virus, because when I was in UK, as Dr. Patil had mentioned, I was with Professor Roger Hull. We had a project where the Southeast Asian RTBV was studied. So we started in Delhi, we started this work. And apart from going ahead with resistance work, we thought of using the promoter. Now the RTBV map DNA is shown on the left side, and cauliflower mosaic virus DNA is shown on the right-hand side. And as I said, CMV35 is promoter, which is shown on that in the encircled. That has been used for last 30 years almost in expressing transgenes in various plants. So the idea was whether the similar promoter in RTBV can also be used, especially for monocots. So that was the thought behind the project. And so we took this area and enclosed by promoter, about 800 base pair, and generated some transgenic rice lines. So this is a somewhat complicated slide. So what you see in the middle is that the yellow line is the RTBV DNA. And what is shown is the TSS. TSS is the transcription start site, where the RNA generation starts, RNA transcription. And then there is almost a 700, 800 base pair region on the yellow, which is the transcribed, but it is not translated. So it can be thought of as a, it can be thought of as an untranslated region. So one of our PhD students, Saloni Mathur, she generated rice plants with this RTBV promoter and various deletions of the RTBV promoter. So various deletions are indicated by those lines, purple and yellow and blue. So what she saw was that the purple line, the deletions, the UD1, UD2, UD3, etc., they showed a expression pattern which indicated that the expression was development specific. So on the left, you see that they are the cross section of rice plants, shoot and root. So 7 days after germination, and then the 30 day after germination. So the purple line deletions, they were showing expression in the, on the 7th day, it was showing expression in most of the tissues. But on the 30th day, it was showing expression only in some vascular bundles. And in the yellow lined sections, which is called C type. Now, C type means constitutive type. That means both in 7 day and 30 day, there was expression in most of the tissues, in both as shoot as well as root. And in the blue lined sections, the blue line is called any type, means non-expressing. It was not expressing at all. Although the promoter is there, these deleted varieties of the promoter were not showing expression. So what I have written on the red colored thing, the study identified a potential negative element, and that was just 137 base pair residing within the untranslated region of the transcript. That means there was a negative element which was preventing the transcription. So overall, we said that the promoter showed development specific and tissue specific expression pattern. And this we filed for a patent because this we realized it was a new type of expression pattern. And this was granted a US patent, and I'm sure you cannot read it. The patent is shown on the left. This was granted in 2010. And simultaneously, we were having discussions with some seed companies. And there was a seed company called Bejo Sheetal seed company. And this purchased this promoter. And we were lucky that they paid us some royalty and all that. But then that is a long story. We don't want to go into that. But here you can see on the back, Dr. M.K. Bhan is standing. He was secretary, I think. Yeah, secretary. And to the right, these are seed company people. Yeah, anyway, so that was a good success story because that was the first time our department earned some money from a product which was developed here. So I want to talk a little bit about the negative element. So we found this negative element, as I said, in the residing in the promoter. And so when we fuse the negative element on the top, you see the 35S promoter driving a Gus gene. And this, when you bombard it in onion or rice callus or Arabidopsis leaf, you can see expression of the Gus gene. But when we have the negative element in front of it, there is no expression. And the negative, then the yellow thing is negative control. That means is some non-specific DNA. When we put there, it was still expressing. So the negative element is not switching off because of its physical presence. It is doing something to the promoter. The negative control is not preventing the promoter. And then we also cloned the negative element in the opposite orientation. It was still able to switch off the promoter. And most interestingly, the negative element upstream to the promoter, where you see on the bottom, that was also able to switch off the promoter. So on the right, we have some hypothetical models where the promoter is important for the transcription machinery. And when you have the negative element, it is somehow interacting with the promoter and not allowing the transcription machinery to transcribe. And so what we concluded was that this negative element acts as a level of transcription, not at the level of transcript stability. So even upstream to the promoter, it is influencing the promoter. That means it's not influencing the transcript stability, but it is not allowing the transcript to be formed. So I have put a question, potential uses of the negative element. That is an open question. But negative element means it depends upon it is up to the imagination of the investigator what he or she wants to use the negative element for. Because potentially the negative element can be used to switch off anything, any gene. But then we know of silencing, the transcriptional gene silencing. So you can actually silence the negative element and then switch on the gene. So it will probably be a useful handle to control gene expression of in some future, but it is still at the development stages. So I don't know of anybody using any negative element for this purpose, and there's definitely not this one. But this sort of things are there. It is open to scientists to imagine how to use them. Okay, now I will talk a little bit about using Gemini viruses for expressing interesting genes. Of course, we have not really expressed any interesting gene, but this one is a map of Sri Lankan cassava mosaic virus. This is a very, very widespread virus in the cassava crop in our country as well as in Sri Lanka, and nowadays we know it is also present in Southeast Asia. So it is a bipartite Begomo virus or Gemini virus, DNA A and DNA B. In this particular case, Sri Lankan cassava mosaic virus, DNA A does not require DNA B, it seems, in the experimental plant Nicotiana benthamiana. So that's what I've written there. Function of DNA B appears to be dispensable or unimportant in Nicotiana benthamiana. So what one of our ex-students did, Nabanita Gogoi, she replaced the DNA B. There are only two DNA B genes, BC1 and BV1. So BC1 was replaced by GFP on the left, on the bottom part, and on the right, on the bottom part, the BV1 was replaced by GFP. The DNA A was not disturbed, and when you agro-infiltrate this constructs on Nicotiana benthamiana leaf, within 5 days you see fluorescence because of the GFP expression, both in an intact leaf, in just without any amplification, as well as confocal microscopy. So within 5 days, you can start seeing the expression of this protein. And she also quantitated this. And so within 5 days, and it actually increases a little bit at 8 days after inoculation. And the bottom part, what is indicated is the PBI 121 is the 35S promoter. The 35S promoter compared to the 35S promoter, so one of her constructs was expressing at least 3 times more. But this can be further, no further work was done, and this was not even published till now. So if one replaces the DNA B promoters by some strong promoter, this can be further increased. So this is just one example of how one can use Gemini viruses for protein expression. Okay, and now I will discuss a little bit about okra and cassava viruses. So okra virus, what you see on the left top is the okra initiation leaf curl virus, which is also a very important okra virus in our country. And the Gemini viruses, there's something called late promoters. Now these late promoters drive the expression of the coat protein gene, and they are activated by another viral protein known as AC2 or C2. And this is also called TRAP. TRAP means transcriptional activator. So we wanted to see how this system works for SLCMV promoter, the CP promoter, because a lot of information is available in other Gemini viruses, but this one we did not have too much of information. So this is the simplified diagram. So what you see is that the DNA A, it has two promoters, one driving the leftward, the coat protein expression, rightward, which is the coat protein expression, which is the AV1, the red one. And the purple one is the AC1 or the rep expression. So these are in the absence of the transcription activator protein, the promoter is very weakly expressed. But in the presence of the transcription activator protein to the right, what you see is that it is highly expressed. So we wanted to ask the question, can a complete AC2 is required, or a truncated AC2 will also work? Or can the transactivation be seen as a transgene? Or even can another heterologous AC2, not from the same virus, also carry out transactivation? So again, this data I had presented about 2 years back, and this was finally published last year, 2024. So this is the Ukrainian leaf curl virus promoter driving the GFP expression. And on the left, this is in Nicotiana benthamiana, left top. And the right top, this is in okra. And on the bottom, this is in cotton. So this Ukrainian leaf curl virus, I'm sure you know that both okra and cotton are malvaceous plants. And Ukrainian leaf curl virus naturally infects cotton once in a while. So we used cotton as a experimental system to see whether it can drive expression in cotton. And yes, it did. And on the bottom, without the virus, it was expressing at low levels. And on the right, with the virus, it was expressing at high levels. So what basically is showing here is that the Ukrainian leaf curl virus promoter gets transactivated by the virus itself in Nicotiana benthamiana, which is an experimental system, in okra, which is a natural host, and in cotton, which also can be thought of as an experimental system. And this activation is at least 3 to 4 fold, which is shown on the right, with and without OEL-CUV. And this one is the Sri Lankan Kasav mosaic virus CP promoter in Nicotiana benthamiana. So again, something similar what we have seen in Ukrainian leaf curl virus is happening in for Sri Lankan Kasav mosaic virus. This one has not yet been published. So we have without just the virus on the top left, you see very low expression of the promoter. But with tomato leaf curl New Delhi virus, which is a different virus, if you put the AC2 together, all this is transient expression. So the expression level increases at least 3 to 4 fold. And with Sri Lankan Kasav mosaic virus, it goes little more further. And with tomato leaf curl New Delhi virus intact, it goes still further. But when compared to 35S promoter, it is slightly lower. 35S promoter is driving maximum amount. And on the right, what you see is that we have used the truncated AC2 gene of Sri Lankan Kasav mosaic virus. So even the truncated AC2 gene, which is shown on the top right, the AC2 gene of Sri Lankan Kasav mosaic virus is 135 amino acids. And the truncated gene is 100 amino acids. Even the truncated gene is able to transactivate the coat protein promoter of Sri Lankan Kasav mosaic virus. And on the bottom right, you see the amino acid alignment of the Sri Lankan Kasav mosaic virus, tomato leaf curl New Delhi virus, and Ukrainian leaf curl virus AC2. So you see there is a lot of similarity, but there are some differences also. So what we found is that these two, the Sri Lankan cassava mosaic virus promoter can be transactivated by both the tomato leaf curl virus as well as Sri Lankan cassava mosaic virus. And another student of ours, Rohit, he generated transgenic Nicotiana benthamiana, where this particular reporter gene, Ruby. Now, Ruby is the name of the chemical compound is beta-lane, which is a derivative of tyrosine. It's a natural product. And there are 3 genes which can be inserted into the system, which converts tyrosine into beta-lane. So on the right, what you see is a Nicotiana benthamiana leaf in which the Ruby gene construct is driven by 35S promoter. It was just agro-infiltrated. And you see the red color coming up in 3 days after inoculation. And this one here, what you see is that the transgenic Nicotiana benthamiana driving the Ruby gene under the CP promoter of Sri Lankan cassava mosaic virus. So without anything, when you have that mock on the left, there is no expression of beta-lane. But in the presence of AC2, it is expressing. And even in there are some mutants of AC2 stop 1, start 1, it is not expressing. But the mutant AC2 stop 2, it is expressing, which means that the reporter gene is actually a transgene. And this is being transactivated by the AC2 protein infiltrated through agrobacterium. And on the middle, you see that this is being activated even by SLCMV inoculation on the top compared to the 35S AC2. And on the right, we are showing the transgenic expression in tomato. We generated some transgenic tomato lines where the Sri Lankan cassava mosaic virus promoter is driving the expression of GFP. And this shows the expression, the transactivation in the white bars compared to the gray bars without transactivation. So what I've written on the bottom is that several uses of inducible promoters can be envisaged in the future. Very few publications have come out on using inducible promoters, but it's up to the imagination and resources of the investigator to use these sort of promoters. So this is just one example, but there are many viral promoters which are inducible by various viruses or other systems. Okay, I will spend a few more minutes with discussing virus-induced gene silencing. I'm sure most of you have already heard about it, and many of you have probably used it. So these are transient gene silencing tools which can be used, which uses modified viral vectors and takes advantage of the inherent RNA interference-based viral antiviral immunity in plants. It bypasses the need for genetic transformation. It can silence multiple genes and results can be obtained in a few weeks. So we first started working on VIGS using the RTBV. And again, I don't want to go into the technicalities of this. This basically shows the mechanism of virus-induced gene silencing, which is based on RNA interference. I don't want to spend time in explaining this. But next thing also is a very general slide, which means that after VIGS, you can use it to analyze various properties of the silenced genes. For example, the phenotype, the metabolic contents, the environmental microbial resistance, or gene expression, other gene expression and transcriptomics, and etc. etc. So VIGS is a starting point for studying gene function, you can say. So we used, first we did this work on RTBV, and RTBV, we developed a gene silencing vector for rice derived from RTBV. And here there were some modifications. For example, a maize ubiquitin promoter was added, etc., etc. Again, these are all published, so I don't want to go into details. Yeah, so this is the picture of what actually we do. This can be injected in the rice plant and the crown region of the rice plant. And this was published in 2010. And we started seeing gene silencing. The first was with phytoin desaturase gene. So the leaf on the right is the inoculated one where phytoin desaturase is silenced. So you have white streaks coming in the leaf. And the PDS transcript level also started falling within a few days. So within 25 days, actually. So it's a very good system of gene silencing in rice plants. And this shows the what we have shared this WIGS vector with various research institutes across the world. So from starting from year 2011 till to 2023, when I sort of retired. So we have given it away to many research institutes and universities across the world, both in India as well as abroad. And it was also, we had applied for a patent, and it was the national patent was granted sometime in 2015 or 16, which is shown on the right. And we have published some more articles on standardizing the silencing thing using in rice with this vector. And what is more interesting is that people have already started using this system. The left on the top is a paper from China where they have silenced some genes in wild rice, Cynodon dactylon and Zoysia japonica, using this system. On the top right is a paper from NIPGR where they silence some rice genes for leaf length, etc., which we were not even aware of. I mean, it was totally independent of us. And on the left bottom and the right bottom, we collaborated with people from PAU and NICPB and IRI, NRCPB, I think, for functional validation. The right bottom one appeared just a few months back. Dr. Kalyan Mandal and etc. So, okay. What about vegetable species? Again, I don't want to, I'm sure here there are experts, but various viruses have been used to silence all different types of genes in a large number of vegetable crops. But some viruses I've just listed: the tobacco rattle virus, then apple latent spherical virus, tobacco ring spot virus, etc. And these are specialized for certain classes of certain families of vegetables and fruits. But there are many challenges, of course. And I've given this reference, which was published recently, and we also have a publication in 2022 where all these things have been discussed. Finally, I think the last few minutes, I will spend some time on virus-induced gene silencing. And this mostly this information I've taken from this recent report, which I've listed there in 2025. I think it was published in July. Yeah, June, sorry. So this person, Voitas, he's a pioneer and he's been very active. And not only he, but also our Dr. Dinesh Kumar and many others are very active in this field. So for most gene editing projects, you have the expression of the sgRNA and the Cas9 or the nuclease together in the target plant. Now, if you can use viruses for this thing. Most people use, because viruses cannot carry a lot of extra DNA. So most people use the virus to deliver just the sgRNA. And the Cas9 is expressed in the plant itself. But the idea is that how to avoid tissue culture, because tissue culture, all said and done, there are some experts in tissue culture, but reproducibility of tissue culture is a challenge, especially in crops such as, I don't know, in cucurbits or malvaceous crops and all that. So can virus-induced gene silencing avoid tissue culture or completely? We see some promise. although it is not perfect. So what I've written is tissue culture is one of the most important constraint in making gene editing widely applicable. It can be used in specific crops, but if somebody wants to multiply it in widely different crops, this is promising. Uh, so this will be a huge step towards making gene editing in plants truly applicable over numerous species and important cultivars. So what we have is that normally we have on the right side, what you are seeing is through tissue culture, you generate a transgenic plant which is expressing the nuclease and in most cases expressing the sgRNA. But you can use viruses to deliver the sgRNA in a transgenic plant expressing the nuclease. And so that will give rise to gene edited plant. But you can also use viruses to deliver both the nuclease and the sgRNA. And if it can be delivered in the meristematic tissue, then theoretically you can completely bypass tissue culture, which is the promise. So that is the idea. So what I have written in the middle is gene editing requires generation of large number of events across several varieties of crop plants. So lack of reproducible tissue culture protocols could be a very serious handicap for scaling up gene editing. You can do in some one or two varieties and one or two species, but across dozens of varieties and dozens of species, it's a huge problem. Tissue culture free gene editing promises to be a major technical advance, but as of today, it is still a promise. But it is up to scientists to use the knowledge of viruses for getting into it. Now, one of the biggest challenges is to get the virus into the meristem. So apparently there is a block, there is a barrier of viruses to enter the meristem. There is no clear knowledge what that barrier is. But there are a few viruses which can bypass the barrier. So that is shown on the left. In the middle, what is shown is you can give a key to the virus. That means there is you can attach a motif which will allow the virus to enter the barrier, cross the barrier, opening a gate sort of. Or you can use the virus to break the barrier, basically, I mean, by force. So that's why I've put a hammer there. Or you can put the virus, you can ask the virus to go to a non-meristematic tissue and then convert the non-meristem into a meristem. So these are probably the 4 ways by which you can ask the virus to get into the meristem and deliver the sgRNA and the nucleus, if possible. So these are the 4 challenges which I have, and in fact, I have taken it from this, I think the next, the same publication. So challenge 1 is how to increase the virus entry into meristem. So either use a meristem entering virus, and there are a few viruses which can do that. Barley stripe mosaic virus and tobacco rattle virus. There are very few and they have low cargo capacity. Or attach a mobile RNA motifs, and most common example is modified flowering locus T. It is called FT. Now, flowering locus T is a protein which moves within the plant. So this can be used. This can be attached to the viral vector, and then it will allow the virus to enter. Or a tRNA-like motif. Now, the problem is these motifs are not well defined. So a lot of research has to be done on it. The third strategy is to down-regulate the defense gene or try to break the barrier. So you can have an antiviral defense. You can weaken the antiviral defense by silencing some of the defense genes, by expressing RNAi suppressors, which are viral suppressors, which are designed to down-regulate the RNAi defense. Or you can use expression of movement proteins, or some people also use cold temperature. to allow the viruses to enter meristem. Or finally, you can convert non-meristem into meristem. So there are tissue culture tricks like cytokinins or inducing the non-meristem to become a meristem. So all these are possibilities, and there are people who have, there are publications on it, but these have to be applied more widely. Second challenge is how to increase the cargo capacity, because if you want the both the viral vector to deliver both nuclease as well as the sgRNA, the cargo capacity has to be modified. So some viruses, for example, barley striate mosaic virus and tomato spotted wilt virus, Sonkus yellownet virus, they have large carrying capacity. And they can easily carry Cas9. Now, Cas9 has a size of more than 4 kb, but only some viruses can carry that. The second strategy to increase cargo capacity is to use smaller nucleases. So Cas9 is quite a large nuclease, about 4.5 kb. But fortunately, there are some alternate nucleases, Cas12F, TNPB, etc., which are much smaller. And interestingly, one of the TNPBs has been discovered in Indian, Cuttack Central, this called National Rice Research Institute, Dr. Mullah. So this was published in 2016 or 17. So that's it has been used in rice, but not as a virus thing, but it is a much smaller nuclease. The third challenge is how to increase the editing efficiency. So one can either increase the entry into meristem, which is shown on the left, or design a better nuclease, or design a stronger promoter. Now, these are all possibilities, and this probably requires multi institutions and people with various expertise, gene, protein engineering, etc. Everything is possible. So this is what these are some of the viruses, viral vectors which have been used. So BYSMV has been can carry both Cas9, sgRNA, as well as a red fluorescent protein. The size is quite big. BYSMV is almost 18 kb. It is an RNA virus. PVX can carry Cas9. Tobacco ring spot virus has 2 RNAs, and the second RNA can be used to carry Cas9. TRV and BSMV have both, TRV has 2 RNAs and BSMV has 3 RNAs. They can, TRV has been used to carry one of the smaller nucleases, TNPB. It's actually derived from a transposon. And these are nucleases which are directed by RNA. Of course, it's not just a normal nuclease. And on the right, you see the nucleases which have been used. Cas9 is about 4 kb, but then these are the small ones, IS-DRA2 and IS-MYU. These are about 1.5 kb in size. So, and on the right, you have the table which shows the families of the viruses which have been used, families and genera. And so there are only a few, as of today, there are only a few examples of completely tissue culture free heritable gene editing using virus-induced gene editing. One is in, well, there's only one crop, which is wheat. Triticum aestivum has been used, and this is a very recent publication in 2025. Nicotiana alata has been used. This is fairly old, 2015. And then there's one in 2024 in Nicotiana benthamiana, and one in there are some in Arabidopsis in 2025 again. So, so that is the present scenario. So I can conclude by saying that plant viral promoters can become important sources of tissue-specific inducible gene expression platforms. Virus-induced gene silencing is an important tool, and it it is a very powerful tool to uncover gene function. And finally, gene editing using viruses is a promise. There is definitely a promise of making gene editing completely tissue culture free. Finally, I want to acknowledge some of these students and postdocs have specifically worked on promoters and the transactivation. So Basanta Bora, Nabanita Gogoi, Fawzia Zareen, etc., etc. And funding agencies, we received a good amount of funding from European Commission. That was early 2000s. Then DBT and BIRAC also funded some of the work, and JC Bose Fellowship was used. And currently, I'm getting INSA Senior Scientist Grant, which is basically to popularize science and do some very basic research, not much. So I think, thank you, and I'll be happy to answer any questions if you have.
It's open for discussion. Friends, I'm sure you all have heard about cauliflower mosaic virus 35S promoter, which is globally used. And rice tungro bacilliform virus also belongs to the same family of virus, but different genus. That is colimovirus, and this is the Badna virus. So their promoter nature are similar, but to create diversity in usage of promoters and have a lot of applications for tissue-specific expression and all. So this is one classic example where this has been employed and also commercialized. And you have learned about various technologies wherein viruses can be employed as Trojan horses for gene silencing and gene editing technology also, where especially in the crop plants which are not available for tissue culture and transformation. So I'm sure it has given a lot of knowledge to all the people. Sir, there were 2 rooms that have been occupied. Here we have only scientific staff, the scientists, and in another room we have research scholars and students. And this was also telecasted live on the YouTube. I'm sure many would have enjoyed this talk on YouTube. Now this is open for discussion. Anybody has any questions, clarification, are free to interact with sir.
Sir, good morning. Myself Chandrasekhar, sir. Did you identify specific cis element which made transactivation of the viral element, viral promoter?
I think we are doing some work. Well, currently I am superannuated, so I won't say we are doing, but there is some work which went on in our lab regarding the Sri Lankan Kasa mosaic virus promoter. We have not really identified any element, but we have sort of, that promoter is about 300-400 base long. We have managed to shorten it and show that the shortened version also functions. So now it is up to the bioinformatics people to identify potential elements, and then one can mutate them and see whether they really affect the transactivation or not. But these elements are generally known in across Gemini viral promoters. There's a lot of work in Gemini virus promoters, tomato virus and squash leaf curl virus. There's a lot of work from China.
So why I asked this question, even 35S promoter also it's from virus. So does it also has got such kind of nature?
35S promoter, a huge amount of work has been done on it. I cannot give you details about what is known, but yes, there's, in fact, people use double 35S promoter sometimes, and but it depends upon the host plant and sometimes tissue types also. So we really cannot say. It's a big question. Yeah.
Sir, even if we use such virus promoters, in which kind of approach will be more useful? What do you think, sir, for our guidance? Means
no, it depends upon
susceptibility factors or any other like
so that is very important. So the investigator has to first decide what should be the tissue where the target protein or gene has to be expressed, whether in all tissues or in certain tissues. For example, resistance, I can talk about virus. Resistance against viruses. Viruses enter through certain, phloem elements or leaf, etc. So it should be expressed maximally in those tissues. In general, these specialized proteins should not be expressed across the plant. That may have a negative effect. So these things have to be considered. And the RTBV promoter, the rice virus promoter, that is a tissue-specific promoter, at least some of them. The cassava virus promoter, we have not checked whether it is tissue-specific or not. But yeah, it's an open question.
Okay, sir. Thank you, sir.
Sir, I'm Dr. Manamohan. Sir, my this thing is, sir, when you use this either virus-induced gene silencing or expression, so when you go for a sort of spraying or whatever it is, commercially, so then from the biosafety point of view, whether is it can you go for this commercially or how is that?
No,
What is the scenario?
virus-induced gene silencing is a tool for research. So it can be, main purpose is to down-regulate the gene expression and see its effect. So I don't think it is used in the field or as a product. It is a tool for it is a tool to unravel the function of genes. So if that has to be done under laboratory conditions or in controlled conditions, and then you know that yes, if this gene function reduces, you have this phenotype. Then you can go for gene editing or any other thing. So virus-induced gene silencing is not a product, but it's a tool in that sense.
Only for reverse genetics analysis.
Only for reverse, yeah, in a sense that way. It is always done under laboratory conditions or under controlled conditions. Biosafety wise, because it is not done in the field conditions, this is not a major issue as long as it is done within controlled conditions, and there are biosafety guidelines also for this. So most of the virus-induced gene silencing work is done under controlled conditions.
Sir, I am Vagish Babu. Actually, sir, we have the one peculiar problem that we are working with perennial crops like papaya or pomegranate or guava or like this. So actually, tissue culture is a major impediment because we have to do anyway that route for gene editing purposes. So we are all looking out for tissue culture free, any methodology. It's a boon for us because first one generation takes 3 years, 4 years, and we have to normally genomating. It is a recessive resistance method. So at least second F2 or generation, we are able to see our mutated phenotype. So to go to that stage takes 3 to 4 years at least. So your suggestion?
That's what I was saying, that by virus-induced gene editing is a possibility. It's at total infancy. Hardly, there are people working on it, but there are only few, as I showed, only 3 or 4 publications in the last 10 years. Of course, all of them 2024, 2025, few publications on totally tissue culture free gene editing. But it all depends upon the viral vector to be used and whether it can enter the meristem. And but the virus should not be seed transmitted. That is a big problem. So the virus is just an agent to deliver the nuclease at the sgRNA in the meristem. The nuclease and the sgRNA will do the editing and then they will die. The virus will also die. Next generation will have the edited gene. So that is a theoretical possibility, and that's what I showed. And I urge all of you to read that article where Vitas gives all details, how it can be, just ideas. Details mean ideas. It's up to the investigators and the funding agencies, of course, most important, to give resources and the facilities for this. I'm sure within a few years, something other already in wheat it is there. So why not in vegetables? Why not in fruits?
Yeah.
So thank you for the very informative presentation. I'm Dr. Usha Rani. Sir, my question is, the criteria what you have selected for limiting only to the 4 viruses for promoter studies. So we are a big list, but then.
Yeah, no, I did not work much. I mean, we did some work on an RNA virus infecting rice, but I did not work on any RNA virus in other crops. It is just that, no, because our main focus at that time was using the promoters for which DNA viruses are considered to be better. But again, there are many RNA viral promoters also which have been used, but I don't know. Widely.
Those promoted from belong to polyphenol family. Because of their genetic architecture, we have to integrate them in the double-stranded DNA. So that could be one primary reason where the control elements and all could be, matching up well with the eukaryotic system.
Yeah, the other problem is RNA viruses. Again, it's a good question, but not enough information is available. RNA viruses also have promoters, but they are in RNA and they are generally recognized by viral replicases. So if you take that promoter and put into the genome, it may not work. So, but probably there are some, I don't know, I have to see. But generally people use DNA viruses. Cauliflower mosaic virus, 35S promoter has been used for last 30 years. I think the first paper came up in 80s, early 80s, and our RTBV, which we used, is very similar to cauliflower mosaic virus. In fact, there is Dr. Day, NK Day, in Katak, not Katak, Bhubaneswar, that Institute of Life Sciences, ILS. He has used other DNA viruses for similar purpose. Now he's, I think, retired. Nrishingade. I think he is retired now. But he has also worked on other DNA viruses, not cauliflower mosaic virus or rice tumor vesiform virus. So he has also used. So there are several promoters of DNA viruses available for use.
No, the case of banana where DNA viruses are integrated within the banana genome, and under stress conditions they are released and they start replication. So I think DNA promoter elements from DNA viruses are well adapted with the eukaryotic system, because there the DNA is the heritable element. So that's the ease. So RNA viral promoters maybe for E. coli expression and all, they could be okay, but not for higher eukaryotes.
I am Dr. Shivshankar. Sir, with respect to VIX, virus-induced gene silencing, usually we have seen that it is a transient. For example, PDS, when Nicotian benthamianum is induced using PDS genes, usually we see bleaching, but transient, but afterwards plant will recover. So how to make it permanent? It should not recover back. Is it possible?
No, it is, as I said, virus-induced gene silencing is just a tool. So when it starts, it is always transient.
Yeah.
It is never, I don't think it is ever permanent. It's always transient. But once it starts silencing, then one can do a lot of investigations, like what is the effect of the silence gene down regulation. I think I showed you in one of the slides that you can do metabolite profiling, you can do transcriptomics on the silenced tissue, maybe for 2 weeks, 3 weeks. But at
Yeah,
that
that's
time,
right.
you can see what are the effects. So you can see how it interacts, what are the networks, what are the interactome. So that is the whole idea. You cannot make it permanent.
No,
That's
even virus-induced gene editing also will be transient only then?
The expression will be transient, but once the sgRNA and the nuclease are there, then they will go and edit in the nuclear, the gene.
So that is for proof of concept establishment.
Yeah, that's
Ah.
We have to
go for normal processes like either DNA based or because once the gene editing is done, then virus is not required. Then it is permanent. So, but it is tricky, as I said. But most of the people for gene editing, they are expressing the Cas9 and the sgRNA, I think constitutively. They are always expressing, and then they are editing the gene. Then they are separating the Cas9 by breeding. They are separating. So tissue culture is taking time. Then the removal of the gene is taking time. So this will shorten. But it is all promise. So only in wheat and in Arabidopsis it has been. Okay.
Yeah, now I request our director, sir, to give his remarks.
Okay. Students.
Difference.
Okay.
They can always ask Dr.
Yes, okay.
Patil or
No problem. So you can also take your email address also,
Yeah.
if it is possible
yeah,
for
yeah,
you.
sure,
Sir.
sure.
So thank you very much, Dr. Gupta Sahab, Dasgupta sir. It is really, because I was, in the meantime, I have some other meetings, so I went there. So sorry to take all the things in together. But it is mentioned very things, many of these informations, what we got it from your particular presentations is definitely my students and also all of the scientists also here. And they can take it up and anything they need to have some clarifications, also they can also directly, they can contact you. Also through Dr. Patil, also they can contact you. But it is again mentioned that virus is really important to all of our crops. Particularly today also we are discussing in marigold, new viruses also coming in the way of what we have. So again, I'm telling the flower crops also, you have a lot of viruses are there. We have not worked together in this particular aspect in virus is concerned. Only you are supposed to have work in some of the vegetable crops and some fruit crops, but the fruit crops also not been done so far so vigorously. But again, we have to take many of these viruses, which is also done. But as you mentioned, one of the important thing that particularly the tissue culture free virus induced genome editing, which is very important, and also as you mentioned. And just a few days back, what I've seen the in somatic cells also can be used for the reproduction point of view. We have a very good in the nature says come. Taking the somatic cells and then chromosome and put it in the fertilization system. It also give the fertilized eggs, which is again important nowadays. What is possibility, impossible is not there. So from somatic cells, so we can also take the reproductive cells and go for fertilizations. So that also is coming in the nature's last 2 days back. It is there in the nature genetics. So that sort of things also comes in the way of what we have, which is totally impossible, but it became a possible. So that what you are told also can be possible. These are things which can also take off in the future. I think this is very important topic, and you have given a lot of inputs to all of us. We'll also take up with our scientists and also our advocates. So I thank to you all, and also to Patil. He has also taken care of this type of activities here. I think we can have a better for virus resistance to do more aggressively, because as I mentioned, many crops we have that problem. So definitely we'll also take your help, your guidance, so that in the future also we can take up many of these issues. So thank you on behalf of my institute. I think we'll again have a work with you, sir. Thank you very much.
Thank you, sir. Yeah, first of all, I would like to profusely thank Professor Dasgupta for taking time to visit IIHR and delivering a wonderful talk. And I thank our respected director for taking time to participate in this and overall making sure that everything is fine despite his very busy schedule. I thank all the heads of the department, all my colleagues, students, research scholars, and all those who are online on the YouTube for taking their time out from the busy schedule and participating in this classic lecture and contributing for all the discussion. And we look forward for interaction in a virtual mode later. Thank you, one and all.